It's undeniable that the microbiome is one of the hottest topics in the last years. This has been catalyzed by a huge international effort, for instance from the Human Microbiome Project. And what is more intriguing is this connection between the gut microbiome and human health. Also interesting is that it's not just the diversity and composition of the microbiome, but also the dynamics and evolution of the microbiome, and this has been expressed in different papers in the last years. And this matches pretty nicely with this promise of personalized medicine. So the idea is very simple. You start with some experiments in which you are putting some bacteria into competition, and then you plug those experiments, those data, into a mathematical model in a computer, and then you find the interaction network. What is interesting is that the promise is that now that you have the interaction network for this specific patient, you can somehow intervene and try to solve some health problems. For instance, imagine that the yellow bacteria in this diagram is the dangerous one, then you can see that maybe introducing more green bacteria or some food related to the proliferation of this green bacteria, then you can target the yellow one in an ecological way. In this presentation, we borrow this sentence by George Box saying that all models are wrong, but some are useful. And we give 3 compelling arguments why this whole enterprise might be doomed, and also highlight the way in which we could fix this conundrum. So let's go back to this project and focus on the mathematical model. We're going to talk about the generalized Lotka-Volterra, but the problem is much more general. However, it's undeniable that this model is at the center of many of the arguments proposed in the literature in the last years. So our 3 concerns are related to 3 questions. The first one is, can we estimate the parameters of the model even with perfect information? The second one is more realistic, is what if data are noisy or scarce? And the last one is more profound. So maybe we can feed the data, maybe the data is not that noisy, but can we interpret the fitted parameters in a unique and concise way related to the microscopic dynamics of bacteria? These 3 questions are related to different concepts that we are going to develop in the following slides. Let's start with the first question. A model is structurally identifiable if its parameters can be uniquely estimated by fitting the model to experimental data. Let's take a simple example. Imagine a population that has a simple decay, but this decay depends on 2 mechanisms encoded in these parameters lambda 1 and lambda 2. This model is not identifiable. Why? Because if we increase the value of lambda 1 by a given factor and then we decrease by the same ratio parameter lambda 2, then the model remains the same. So this model is not identifiable, as we were saying. The good news is that in a recent paper it has been proved that the Lotka-Volterra model is actually identifiable in this sense. However, we have to be careful with this interpretation, because this result comes from the idea that you have the whole dynamics of the model. But if you take account of the number of parameters that you have, and you have, for instance, n parameters of the single growth R and n squared parameters of the interactions beta, then this cannot apply for the steady state, because in the steady state you only have one equation for each of the parameters at the steady state. So we just have n data points for n times n plus 1 parameters. So identifiability is fulfilled if we have a dynamical time curve, not just data coming from a steady state. Let's go to the second question. So can we actually do with real data? And some papers say, okay, you can actually. You have to be careful with the sampling, and you have to be careful with the noise and the way in which you design the experiments. And if you do that, in principle, you can extract this network of interactions, and you can say that a couple of species have some relationships like mutualism, commensalism, and so on and so forth. But let's do a simple numerical experiment. We're going to use the Bayes rule. Actually, we're going to try to infer the parameters coming from the data, and this comes from this simulation in which we generate the data coming from the parameters. So this is the simulation. We have 3 populations, very simple dynamics. We add some noise, and now we run the inference model. We are not going to give the details about that, but we are using Monte Carlo Markov chain to do the inference. So this is the posterior predictive trajectories, and you can see that the trajectories pretty much match the data within this uncertainty coming from the noise. But here comes the intriguing thing. So we have used a Monte Carlo Markov chain with an initial set which is far away from the original parameters. And some pathologies appear. So let's take a look at some of these panels. Here you can see that we are forcing the parameters R1, R2, and 3 to be positive, but you can see that somehow the distribution wants to go to negative side of distribution. This means that the inference is exploring parameters that are not realistic, but we are not allowing to do that. What is the consequence of that? That in some situations like this parameter, you can see that the mode of the distribution is close to this vertical black line, which is the original parameter plugged into the simulation. But the problem is that we have a probability that the parameter takes different values. The red line separates the negative from positive values, and you can see that almost 50% probability of having a different sign. You can see another situation here. In this case, we have the right sign. We do not have problems with negative values, but you see that the estimation, the mode of the estimation, is far away from the original value, which is, again, the black dashed line. And again, you can see here that we have a non-zero probability that the sign of these couplings are different. In this case, populations 2 and 3 can change the way in which they see each other. So the prey has a probability of being a predator, and the predator has a probability to be a prey. So this whole promise that, in principle, extracting data from the fitted parameters, and then you have the network, and from the network you have the interactions, and from the interactions you know the right treatment for this bacteria, is not accurately produced even with a very small system with 3 bacteria and under controlled situations. So you can see the problem. With just 3 populations, we have highlighted that just adding noise to the data can introduce some uncertainty in the predictions of the parameters. So imagine that you have n populations and you extract the data not just from one experiment, but from many experiments coupling different bacteria one by one. So this promise that you can estimate effective parameters using experiments and models is not that clear. Another possibility is extract the interactions doing all the possible experiments, so starting with 1, 2, up to 7, for instance, interaction between parameters. In this case, you can actually know the real microscopic interaction between bacteria. The problem is that they have been done in a steady state, so you cannot plug those steady state parameters into the equations that we have seen before. Finally, let's address the third concern. Imagine that you have one of these time courses. This is the population of a given bacteria at different times. And you ask this honest question, how many pieces of significant information can you extract from this figure? Let's count. So you have the steady state, the peak to steady state, the time to the peak, and the duration of the peak. Okay, so in the case of n populations, you have an upper bound, so at most you have 4 n parameters that you can estimate. If you're lucky and you see the curvature of this part, you can say 5n. So let's draw this curve. So the orange one is this estimation of 4m pieces of information for n populations. The green one is 5n, and n times n plus 1 is the number of parameters of the Lotka-Volterra model. You can see that beyond 3 or 4 populations you are doomed, because basically you have more parameters than actual pieces of information relevant to the model. And this can explain why the Bayesian inference cannot recover back the parameters that we introduced in the simulations. We were playing with 3 populations, and we are in this threshold in which basically we don't have so much information. Now the question is, can we do better than this quick and dirty estimation? And the answer is yes, we can do this systematically, and this falls into the realm of the so-called theory of sloppy models that has been developed by James Sedna and collaborators. And despite the fact that the mathematics are really complex, let's do a very simple experiment. Imagine that we have a population that has some decay, and try to model that with 2 parameters, θ1 and θ2. You can see that different parameters provide different curves, which are these dashed gray lines that you see there. Now, as I was saying, we don't have full observation, we have just 3-point observation, which are related to these vertical lines. So imagine that those parameters and measure at those give these 3 points. If we generalize this, and we can extract different estimations for the model. So basically we have a theory, which is the sum of 2 exponentials, at different parameters correspond to different 3 points, and this produces this manifold, which is the space of all potential predictions of this model. You can see that this manifold is not regular, so you have some peaks in the surface. You also have some curvature, as you can see here. So different parameters are at different locations of the surface. What is central to this theory is that this manifold has some boundaries, as you can see here. So this boundary corresponds to this parameter going to 0. This boundary corresponds to one parameter going to infinity. And this boundary corresponds to both parameters being the same. So different boundaries correspond to different limits of the theory. So you can see that the whole original model can split into 3 different models, which are simplified versions of this one. Actually, if for some conditions in which these parameters also go to 0 infinity, then the relevant model are simple constants. So the population is at a steady state with levels 0, 2, and 1. So this is the end of the summary. So let's do some numerical experiment going back to our problem. We have 4 populations. We are going to do some deterministic dynamics, and we're going to do that free of noise and uncertainty. So basically we have these circles, and the circles follow this smooth line, which is the prediction of the model. If we don't remove any model, so we have all the parameters, you can see that the solid line goes through the circles as expected. And now we apply this methodology of the sloppy model. So we do that. We have removed one parameter, but you can see that the solid line is still above the circles. Let's remove two parameters. This is another manifold, another boundary of this manifold we're mentioning, and this works pretty nicely. And let's do that on and on and on. And then we remove 9 parameters, and you see that the predictions now are not that good. You can see that the orange line is not captured pretty well at the beginning of the simulation, but this is an artifact. Actually, if we start all over again, but instead of using 4 populations and 20 parameters, we use just 3 populations because we think that the orange one is a slave to the other 3, then we can see that the other lines, the blue, the red, and the green, are captured pretty well. And we have reduced from 20 parameters to 7 parameters. But what is more interesting is not just the reduction itself, it's the idea that we started with a fully connected network. In principle, all the bacteria interact with all the other bacteria, but in the end we ended up with a higher sparse network. So basically only a few of those parameters are surviving. As you can see here, if we remove the diagonal ones, basically we have just parameter beta 1, 2, parameter beta 3, 1, and parameter beta 4, 1. So basically only we have this interaction between these populations. Another consequence of this experiment is that this estimation that we did before, that we say, okay, we have 4 pieces of significant information and 4 populations, so in principle we have 16 parameters, but we end up with 7 parameters. So this quick and dirty estimation is just an upper bound. We can repeat this to another situation in which you have oscillation. This is a typical Lotka-Volterra simulation. And again, you start with 3 populations of 12 parameters and end up with 2 populations and 5 parameters, and the other population, the green one, is a slave to the other 2. So at this point, you might be thinking, okay, is this a defect or limitation of the generalized Lotka-Volterra? And some authors have thought the same thing. So some authors have proposed that maybe we have to increase the complexity and not just limit to the population of bacteria, but also include some resources. And then the competition is not just through this interaction matrix that we were mentioning before, but it's a competition for resources, and some of them share the same type of resources. But the problem is still the same. If you go back to this estimation of how many pieces of information here, the thing is that the sloppiness is a matter of actual information in the data, not just the model sophistication. So if you increase the number of parameters, adding this interaction between resources and bacteria, you are actually making things worse. So are mathematical models useless? And the answer is no, luckily. So as one of the authors of the presentation has shown recently in this paper, we have to change the question. It's not just about trying to infer the network of interactions. We cannot know what is going to happen at the microscopic level, but we have to think about correlation patterns. We have to think in ecology as a kind of distributional problem. In this sense, we have to ponder what answers can your model provide. Another possibility would be to invoke the artificial intelligence, but we have a problem here. So instead that we replace this mathematical Lotka-Volterra model or variants of that with some artificial intelligence, machine learning, or whatever. The problem here is that we have a black box and we have some problems of interpretability. And the reason why these models cannot work in the end is because maybe they can accurately predict the network of interaction, but this network is an effective one. Because again, the number of pieces of information relevant in the data is not that large. So to finish the presentation, just 4 messages. The first one is that you should do the math before fitting, and remember that the steady state misses the point because you have a limitation in the number of parameters that you can extract. The second one is that you can fit, but you have to be careful with interpretation. So some of the parameters that you are inferring could be compatible with negative values or positive values of those parameters. So you should go Bayesian and have an idea of the uncertainty of the prediction. The third idea is that sloppiness comes from information in the data, not model sophistication. So doing things more complex are not solving the problem, but increasing actually the problem. And the last message is more profound, is that models are not wrong, but basically we are trying to do some macroscopic inference about microscopic data, and you cannot do that. So you should ponder what answers can you ask your model. And that's all. Thank you very much.